Imagine holding a key to the past, a glimpse into the very cells of a creature that roamed the earth tens of thousands of years ago. That's exactly what scientists have achieved by extracting ancient RNA from the body of a woolly mammoth named Yucca. But here's where it gets controversial. Could this breakthrough not only reveal how these majestic beasts lived and died, but also unlock secrets of long-extinct pathogens that once plagued them, and possibly us? Let's dive in. In 2012, paleogeneticist Love Dallin from Stockholm University had a jaw-dropping moment in a lab in eastern Siberia. His Russian colleagues led him to a room where a remarkably preserved woolly mammoth lay before him. It doesn't look like it died yesterday, but you can't believe your eyes because it's so well preserved, Dallin recalls. It's a kind of holy hell moment when you see this. Yucca, the young mammoth, had been thawing out of a permafrost cliff near the Siberian coastline, her body frozen for nearly 39,000 years. And this is the part most people miss. While DNA, often called the recipe for life, has been studied extensively in mammoths, RNA is the unsung hero that translates this recipe into action. RNA molecules act as messengers, instructing cells how and when to build proteins. Unlike DNA, which remains largely static, RNA is dynamic, changing rapidly in response to factors like stress, feeding, and infections. This makes it a treasure trove for understanding what was happening inside an organism at the moment of its death. But extracting RNA from ancient samples is no small feat. RNA degrades quickly, often surviving mere minutes or hours, not millennia. It felt like a very high-risk project, Dallin admits. It seemed like a completely crazy thing to try to do. Yet, against the odds, Dallin and his team succeeded, publishing their findings in the journal Cell. They extracted RNA fragments from Yucca's muscle tissue, revealing insights into her slow-twitch muscle function and stress responses. Interestingly, some RNA suggested Yucca was under stress at the time of her death, possibly from being chased by cave lions or another cause. Here's another twist. Despite being initially identified as female based on visual inspection, genetic analysis revealed Yucca was genetically male. In theory, Yucca could have developed as a female, Dallin explains, but more likely, critical morphological parts were missing during the inspection. Boldly, this raises a question. Could this technique help us study ancient RNA viruses? Maria Avila Arcos, an evolutionary genomicist, points out that many deadly pathogens like Ebola, COVID, and influenza have RNA genomes. If we want to understand their evolution or how these viruses impacted populations in the past, we need to recover RNA from ancient samples, she says. This opens a door to not just understanding mammoths, but also tracing the history of diseases that still affect us today. While the study is groundbreaking, it's not without limitations. Manasa Raghavan, a paleogeneticist not involved in the research, notes that the success relied on well-preserved samples from a pristine Arctic environment. Applying these techniques to specimens from temperate or tropical regions where preservation is poorer remains a challenge. Yet the implications are staggering. You're actually seeing processes going on inside the cells right around the time it died, Dallin says. And these processes have been frozen in time for 40,000 years. This isn't just about mammoths. It's about rewriting our understanding of ancient life and the forces that shaped it. Now, we want to hear from you. Do you think this RNA breakthrough could revolutionize our understanding of extinct species and ancient diseases? Or are there ethical concerns we should consider when delving into the genetic past? Share your thoughts in the comments below.